On behalf of Aditya Jyot Foundation for Twinkling Little Eyes, I welcome you all at first Aditya Jyot, Dr. APJ Abdul Kalam Public Endowment Oration by Dr. Takeshi Itawa today. Aditya Jyot Foundation for Twinkling Little Eyes is a not-for-profit wing of Aditya Jyot Eye Hospital, as named by late Dr. APJ Abdul Kalam in 2005. I would now request Ms. Ashwini Roge, the Senior Optometrist of Aditya Jyot Foundation for Twinkling Little Eyes, to come and throw some light on the foundation.
Good evening, everyone. I welcome you today, and I would like to tell you a little bit about Aditya Jyot Foundation. Aditya Jyot Foundation is a not-for-profit organization started in 2005 with a mission to alleviate the suffering of the poor, neglected population and aims to prevent avoidable blindness through awareness generation, eye screening, and providing access to quality eye care at an affordable cost. We are named by Bharat Ratna Dr. APJ Abdul Kalam, the Honorable Ex-President of India. We run outreach programs in rural and urban slums and tribal areas, providing eye care to those who cannot afford it. We have numerous projects focused on creating awareness, screening, and providing comprehensive eye care services to prevent avoidable blindness. Some of our activities include the CREAM project, which stands for Children's Refractive Errors, Eye Ailments and Management Project, which basically involves screening school children and providing free eye care services and management of diseases. AJ Drums, which is a diabetic retinopathy screening and management program, which basically provides diabetes screening and management services to the slum population in Dharavi and the suburbs of Mumbai. Pearl Study, which is a pilot testing of which is a pediatric eye anomalies, refractive errors and traumatic lesion study, which screens the slum population, children between the ages of 4 to 15 years, and provides free eyeglasses and other management services. Apart from that, we aim to make the roads and roads of the city more safe by providing eye care services and screening programs to the drivers, truck drivers and public transport drivers in Mumbai. Aditya Jyot Research in Vision and Ophthalmology, which is AJRO, which is the R&D unit of Aditya Jyot Foundation for Twinkling Little Eyes. We conduct research through epidemiological studies. So basically this is the diabetic retinopathy program, where we have a team of trained community health workers who do door-to-door survey activities in the slums of Mumbai. The data that is collected by them is uploaded, is recorded and used for research purposes. Research through genetic lab. So basically this burden of blindness due to inherited retinal diseases, as we know, there are several diseases, genetic diseases, which can hamper the vision and affect the eyesight. We aim to create DNA and RNA repository by studying the genetic profile and identification of genetic markers of diabetic retinopathy, age-related macular degeneration and Stargardt's disease. The methodology is basically we would be studying the clinical and phenotypic profile of these diseases and the gene and protein study. Samples that we use for this study is the blood, the tear, vitreous, aqueous and the epiretinal membranes. And the outcome that we expect from this is basically to develop new treatment strategies which would help clinicians to better diagnose and better treat these diseases. We have also established a basic genetic research lab in Mumbai, in Thane. Under this facility, we would like to conduct eye genetic research with with collaboration with Japan. So these are the basic facilities that we have in our lab right now. This is the plan that we propose to follow. Blood collection and then DNA extraction and RNA extraction from the tissue, and then isolation of DNA, genotyping and exome sequencing. We also are looking at establishing a reading center. The mission is to provide the development of ophthalmic image interpretation, and we aim to understand the treatments for preventing blindness through collaborative clinical research. The primary function of a reading center is to evaluate and interpret the fundus photos, FFAs, of fluorescein angiograms in a standardized, unbiased manner and provide verifiable results. Now, as we know, we are a huge population. It is impossible to conduct screening and provide eye care services to the entire population. So that is where a reading center comes in, where the screening can be done by a semi-trained staff, and those images can be uploaded and examined by a ophthalmologist at a reading after they are uploaded at a reading center. Now, a centralized reading center would help these researchers to perform quality research, lack of ocular epidemiologic studies in various states, minimize variations in clinical diagnosis. So these are some of the photographs. Also, we plan to set up a stem cell lab. Future prospects, we plan to serve as a center that provides for screening of large populations for eye diseases in a cost-effective and standardized manner, serve as the coordinating center for national and international clinical and epidemiological studies, and to allow and conduct novel research to advance knowledge in ocular imaging. Thank you.
Thank you, Ms. Ashwini. I would now request Professor Dr. S. Natarajan, the Managing Trustee of Aditya Jyot Foundation for Twinkling Little Eyes, to come and introduce Dr. Takeshi and also read out the citation.
It's a proud privilege to introduce Dr. Takeshi Iwata, my good friend, and it's written in Japanese, and I know nobody understands except Takeshi Yokoso. That means welcome to India. And Dr. Takeshi first visited India in 1980, he said, when he was 20 years old. And he met several people from Bombay that time, and he decided to come to Bombay. It only took 36 years for him to reach Mumbai. And so he's here with us. He's right now the Division Director, National Institute of Sensory Organs, Japan. And Dr. Iwata graduated and received his PhD from Major University in 1988 and immediately joined the National Eye Institute, the NIH, as postdoctoral fellow. And we have Dr. Professor Kumara Manikvel, who also worked in NEI. And we also have in our audience Dr. Shailaja Tibrewal from Schroff Charity Eye Center, who also underwent a 1-month fellowship in genetics. She's a pediatric ophthalmologist. And in 1991, he returned to NEI to work on the transcriptional regulation of aldose reductase and sorbitol dehydrogenase, one of the major pathways for diabetic retinopathy. He returned to Japan in 1999 and moved to current position in 2007. His research is focused on human retinal diseases to understand the molecular and cellular mechanism of the disease onset. He has worked on normal tension glaucoma, age-related macular degeneration, and a number of hereditary retinal diseases. His research expands from identification of novel disease-causing genes to development of animal models and iPS cells for therapeutic development. When he returned from NEI and started the lab in the Tokyo Medical Center and in National Institute of Sense Organs, he was given a small 10 by 10 room, and now he occupies 10,000 square feet with several research scientists working under him. All with his hard work, he's developed a team, and runs a $15 million lab. And the best part, and he's such a big-hearted man, and I want all of you to give a clap for him, because he has suggested that he will help our foundation to establish the similar $15 million lab in Mumbai. And that's why we have Mr. Nikhil Masurkar here. He also runs, apart from the Entot Pharmaceuticals, the research part in London and in Mumbai. And that's why he's associated with this project. And his recent work on identification of RP1L1 gene for occult macular dystrophy, the Miyake's disease. Miyake was the former director of the Tokyo Medical Center Ophthalmology Department, and he's a Skipens alumni and senior to Dr. Badrinath, as well as he's also a good friend of mine. And he was the one who signed the MOU between the National Institute of Sense Organs and NEI in June 2014 in Tokyo, which I am happen to be there along with John Prakash and Paul Seaving, the director of NEI. So that's how the Asian Eye Genetic Consortium came, and then now Dr. Takeshi is the president, and I am the Secretary General of the Asia Eye Genetics Consortium. And we had a wonderful meeting this morning with people from All India Institute, Shroff Charity, Chitrakot Hospital, and then also our consultant from Singapore, and we had Dr. Kumar. And his work on OPTN gene for normal tension glaucoma and HTRA1 gene for AMD is well known. And when he visited the lab yesterday, he commented that the lab set up by Siddhita, she's a AEGC research fellow who visited Dr. Takeshi's lab, and she started in maybe we are having 1/20th of what he has, the basics of genetic lab, and then we will progress to his level one day. He is now the project leader for the Japan Whole Exome Consortium for Hereditary Retinal Diseases, and over 26 ophthalmology departments, institutions, and universities are involved in this project to collect phenotypic data and DNA samples. And his center has made a database with the storage for all this phenotypic data, and is gracious enough to extend that for entire Asian region for the Asian Eye Genetic Consortium. And except Japan, nobody is using it right now. So first time, the Aditya Jyot will take the lead and use that data, the database, for storing our phenotypic data and also following the DNA studies. He is now expanding this consortium to Asian countries and established an Asian Eye Genetic Consortium with John Prakash, the Executive Director of the International Director of Asia, the National Eye Institute, part of the NIH, US, to share genetic information among Asian countries. And I take this opportunity to present Takeshi Iwata to the audience. Now I have the great honor of reading the citation, and I'll request Mr. Anantaraman, and after I read the citation, to come in the front and give him the... So Aditya Jyot Research in Vision and Ophthalmology, an R&D unit of Aditya Jyot Foundation for Twinkling Little Eyes, presents Scroll of Honor to Dr. Takeshi Iwata, Division Director, Division of Cellular and Molecular Biology, National Institute of Sense Organs, Tokyo Medical Center, Japan, President, Asian Eye Genetic Consortium, on his delivering the first Aditya Jyot Dr. APJ Abdul Kalam Public Endowment Oration on 14th December 2016. And Dr. Takeshi Iwata's research focuses on retinal diseases, age-related macular degeneration, glaucoma, and hereditary retinal diseases to identify the cause and explore the molecular mechanism of the disease onset. His laboratory is responsible for two large consortiums based in Japan and Asia, and the Japan Whole Exome Consortium for Hereditary Retinal Diseases focuses on 36 inherited retinal diseases to identify the disease-causing mutations in Japanese population, with over 30 university ophthalmology departments being involved in the consortium. The Asian Eye Genetic Consortium, an NIH initiative, was started in 2014 by a group of scientists and ophthalmologists interested in exploring the eye genetics in Asian population. and more than 80 members from Asian countries are currently developing a system to share, catalog, and collaboratively work to identify the genetic aspects in eye diseases, signed by Mr. G. Anantharaman, Chairman, Aditya Jyot Foundation for Twinkling Little Eyes, and Professor Dr. S. Natarajan, Managing Trustee of the Foundation.
I would now request Mr. Anantharaman, our chairperson, to please confer the plaque. Now I request Mrs. Shamla Gopinathan, our vice chairperson, to please give away the shawl. I would now request Dr. Radhika Krishnan, the CEO of Aditya Jyot Foundation for Twinkling Little Eyes, to hand over the gift. Thank you so much, everyone. I would now request Dr. Takeshi Iwata to please take over the mic and address us.
It is my great honor to be standing here to receive the first medal under the name of Abdul, President Abdul Kalam. It's very too much for me, actually. But I had a chance to give a talk, a short talk, in 2007 in front of him about Japan and India collaborations. And he was sitting something like this in the stage, and it was a very steep steps. So I walked onto the stage and I bowed to him, and he was smiling and he was waving his hand. So I remember now about that scene, that it was very nice of him to smile and wave at me. So I'd like to thank all the board members, and especially Professor Natarajan, who has been very actively involved with the things that I will be talking in a few minutes. And I have been working in the retina, especially diseases that are highly associated with the genetic defect. So I would like to start with a slide that I would like to thank all the people that did the work that I will be talking to, and mostly the people of the Asian Eye Genetic Consortium, Professor Prakash, Professor Natarajan, and Professor Baird, who is the sort of the member, principal members of this consortium, and many other people are also involved. Also, I would like to thank all the people, principal members of the Japan Eye Genetic Consortium, which is listed here, and also the people that are in my lab who is actually doing the work, and they're right all over this. So I'd like to start with this slide. This is a slide of eye, and many of you don't know about eye, so I will start using this. So you see this a spider eye, the primate eye, the lizard eye. So many of the animals, living things, do need some kind of a organ to detect light. Sometimes this eye will find the food, what they need, and sometimes they want to escape from the predators. So you need some kind of a light detection system in their body to use the light to find something or escape from the enemies. The structure of the eye can be differed by or the animals or insects, and some of the eyes will be very, very simple with just nerves and some cells that will detect light. And like this is the fruit fly eye, and it will be structured something like this. And then in primates, it's more complex. And then we have a layer called the retina, where the photoreceptor cells, where the light will be detected, and the light signals will be converted to electrical signals that will be connected to the brain. So this is the human eye. It's relatively, the lens is small, so the light will be coming through the cornea, which 70% or 80% of the light will be bent. And it will go through the lens to be more focused, and it will go through the vitreous, through the gels, transparent gels, and it will hit the light, will be in the center of the retina called the macula. So the yellow part is called the retina, and this is constructed of 3 layers: the photoreceptor, which detects the light, the bipolar cells, which connect the signal to the retinal ganglion cells that will go out into the brain. The final layer is the retinal pigment epithelial cells. It's a one layer of cells that divides between the capillary, or the blood barrier, and the neurons. So this layer is extremely important, especially for the disease called age-related macular degenerations. So this is the September issue of the National Geographic, and gives you some numbers of how many people who are impaired with visual. So there are 285 million people worldwide who are visually impairment. 39 million people is completely blind. And what is the cause? 40% is a refractive error. So that means that you don't have a good focus in the lens. And some people are with a 30% is a cataract, which is the opacity of the lens. 2% with glaucoma, 1% with age-related macular degenerations. 90% of the patient is in the developing countries, and 82% of the people who are blind are more than 50 years old. So this is the information provided from Professor Natarajan, and it shows a very similar prevalence. So if you're over 7 years old, you will have a prevalence of 20.31% of having a blindness. So it's pretty much almost the same wherever you are in the ethnic group. So what kind of disease are we talking? Cataract, you know that it's opacity of the lens, which is here, and then because it became white, it's not transparent, light cannot go through, and it reflects. And so this is a cataract. But now cataract is relatively easy to remove this and then put an artificial lens. So it's relatively easy now. The major challenge is going to be glaucoma. This glaucoma is a disease that the ocular pressure, inside the pressure can go up significantly high, and that will push, the pressure will push the neurons at the exit of the eye, and it will sort of disturb the cell functions, as you can see up here. Also, another major disease is called age-related macular degeneration. Usually about over 60 years old, 70, 80 years old person will be affected by the geographic atrophy of the retinal pigment epithelial cells or some choroidal neovascularization. That means the choroidal vessel will penetrate into the retina and destroy the neurons. And this is one of the top diseases in the Caucasians, but not in the Japanese populations. So this is the graph that's showing the age and how much prevalence of the blindness. As you can see, as of the age, the prevalence of the blindness goes significantly up. So for cataracts, it's the same situation. For diabetic retinopathy, all the same. And for age-related macular degeneration and glaucoma. However, as you can see, there are white ethnic groups is different. The black, Hispanic, and others, it will be a little bit different among the prevalence. However, as you age, the people with blindness will go up. And this is the prevalence of the Japanese population in the glaucoma. As you can see that in over age 80, you have many of the people with the blindness. Here is another data from India about the prevalence of glaucoma. As you can see, 12.3% is glaucoma, but 47.8% is cataract. So this is the major problem for the Indian populations. This is very interesting data. This is a cause of blindness in Japan, but it's compared between 1990 and 2007. So in 1990, diabetic retinopathy was the number one, and also a cataract here, you can see significant, 12% was a cataract. But in 2007, cataract disappears. So you can go to the clinics and remove the cataract easily. It's not really considered a disease anymore. It's a sort of an aging process. So as the economy grows and the lifestyle changes and the environment changes, the disease will also change in time. And I'm sure it will happen to many other countries as well. So this is the cause of the India. Once again, information from Dr. Nutarajan. And here is a cataract, 62%. And 20% is refractive error, 10% is others, and 6% is glaucoma. Okay? So these diseases has some effect by the genetic factor and also environmental factors. And if the genetic factor is extremely strong, usually the number of patients decreases significantly. We call it hereditary retinal eye diseases. And it can be in the retina, cornea, and lens. And these are very rare, but it's extremely severe. On the other hand, age-related macular degeneration, glaucoma, and cataract have some environmental factor. We still don't know what kind of environmental factors, but sometimes, like for AMD, the smoking, obesity, it can be a risk factor for these. So when you look at the evolution of the humans started from Africa, and they moved out into Europe, and then moved into India, and some of them went to Japan here, and then crossed into Alaska, and then into the South America. And while this humans was traveling thousands of years ago, they have acquired different mutations and then move into these areas. So in each regions, they will have a very specific mutations, specific variations of genome sequence that will be associated with the disease. So if you take one person from this room, one person here, and if you compare the whole genome sequence, there'll be approximately 20,000 to 30,000 sequence differences. So it's very significantly different. So each of them look like similar, but it's quite different. So what is the difference? So we have to a little bit study about the chromosome. This is the human chromosome. We have 22 chromosomes plus X chromosome, and the male will have a Y chromosomes. So each chromosome is, so you put it in pairs. So you have 2 chromosome 1, 2 chromosome 2s, and in chromosome. And one chromosome comes from your mother, one comes from your father, and it comes to something like this. And if you sort of untangle this chromosomes, it will becomes a DNA, which is a guanine, cytosine, adenine, and thymine, a GCAT. These are the 4 molecules that constructs the DNA molecules. And it's compacted into a nucleus in the cells. So, in order to find an association with the gene sequence, we have to read all this sequence of the genome and then find out which part of the sequence is different from the control populations. So the one way is doing is this next generation sequence. You will take this DNA, isolate DNA, you sonicate the DNA into a very small 100 to 200 base pair fragments, little piece of fragments. And you will isolate only the part of where the amino acid protein is coded, which is 1.3% of the whole genomes. And then you do the whole sequence of that. So there are 22,000 genes in our genomes, and you can now sequence all of them in just one day. So this cost of sequencing the whole genome used to be very expensive, but now in 2016 it has hit the $1,000 mark. And with a $1,000 per samples, we can start to do the whole family, whole genome sequence, and still we can continue the research. This is a very simple explanation of the dominant inheritance versus the recessive inheritance. If you have a dominant disease, one of your parents will have the disease, and that gene, one of the chromosomes, will be inherited to his children. And this will continue at the rate of 50% per chance of inheritance. And the recessive, you need 2 parents' allele, chromosomes, that will come into child. So this will only happen at 25% only. So both have to come from the parents. So this is a 25% chance. So if you have 4 children, 1 child may have a disease. But in the dominant families, you will have 50% chance that the patient will appear. So in the disease I'm working on is called the inherited or hereditary retinal diseases. Because the eye is a transparent tissue, over 100 years ago, people would look into the eye and will see different patterns, and they would name it under different diseases. So we have retinitis pigmentosa, Leber congenital amaurosis, Stargardt disease, choroideremia, and all this disease goes on and on, 36 diseases. So when I joined the National Eye Institute at 1988, I joined the lab which identified the first gene for the retinal disease called the gyre atrophy. But now, after 25 years, the genes number has increased to 256 genes. So a lot of genes has been identified, and 12,000 mutation has been reported. However, I will be talking about the Japanese studies that this is not the end. We are now moving into the analysis of the Asian populations. Eventually, we will go into the African populations, and this numbers of mutated genes will significantly go up. Over 300 IM easily go up. So in my Japan, in my institutions, I have associated to 29 universities who would send us the patient information through online, and also a sample. It can be blood samples, it can be saliva samples, and they will be reaching the Tokyo Medical Center, and we will extract DNA and send out these samples out to the companies or the institutions to get the genetic information or the transcriptomes or the proteomes analysis, and we will analyze in our institute which is the real disease-causing problem. So these are the, I apologize for the small letters, but these are all the diseases, the 36 diseases we are working on. And for all these diseases, we need to collect the data from the patient. This can be a fundus photo, a photo of the retina. It can be a fluorescence angiogram. It can be some of the ERG, the electrical pulse that we detect when lighting the patient's eye, and also the sections of the retina, image of the section of the retina called the OCT image, and also the autofluorescence. All this imaging data have to be collected and analyzed before doing the gene analysis. And in order to collect all this information without doing any emails or mails, we have set up an online registration system. The blue part will only be accessed by the clinicians who would put all this information, image information and the patient's images into the systems. And the yellow part is the part that the genetist will analyze the DNA and put their information. And the red part will be the part that we will share the information. So all the ophthalmologists in our consortium don't have to email me. They will just log into the systems, and they will know that their samples DNA has been extracted, the sample has been sequenced, the sample has been already analyzed, and what was the result. So it's a very convenient system, and you need this kind of systems because we are dealing with thousands of samples. So in our systems, we are a little bit unique about this constructing this system is that we have universities A, B, C, D, E here, and they're submitting disease A, B, C, D into the systems. But we now have a disease leader who would monitor the incoming data into the systems, and because they're specialists in particular diseases, they would sort of reject the low quality data and accept only the high quality data that is coming. And this really helps to pull only the same diseases into one pools. And only the ones that are filtered can go through into the analysis. So this high-quality diagnostic system is always needed, and we are probably the only one who is actually doing in the world of doing filtration by phenotypes. So I have talked about this. Okay, so we are currently doing the whole genome whole exome sequence, and we are moving into whole genome sequence next year so that we can detect large deletions and insertions in the genomes. And because we're doing in a family, we not just sequence the patients, this is a recessive families, we will sequence the parents and even the normal brother here. So the filled-in black ones is the patients, and the white ones is the normal persons. So in order to narrow down the responsible genes, you need the cooperation of the parents and even the brother or sister who is not affected. So you need the whole cooperation of the whole family to identify the gene. But if you have a full cooperations, you will able to identify now the real, yes, I'm sorry, somehow the disease name is not coming up, but here we have collected a lot of retinitis pigmentosa, occult macular dystrophy, and Stargardt disease is here. I don't know why it's not showing up, but these are all the diseases that we have been working on. We have sequenced about 600 families, collected about 830 PET families, and then completely sequenced 590 pedigrees. And from this analysis, what we have found is that only 17% of all the families that we have analyzed had a mutation in a published journal, which means that the rest of the families is associated with a mutation not published yet. If this is happening in Japanese population, I think this will happen in Korea and China, and then I think in India. And this was the motivation. This was 6 years ago we found this. And this was the motivation. We need to collaborate with the surrounding Asian countries to identify more genes. So Japanese is basically come from the Korea side, from the north, and also from the south. It's a sort of a mixture. And so thousands of years ago, probably some of the people brought this mutation into Japan, and that's why we need to collaborate and find the source of this mutations. So in 2014, Professor Jyot Prakash of the National Eye Institute and I actually met 1 year before. The reason that we met with Dr. Prakash is that he and I share the same boss, Deborah Copper. She was the deputy director of the NEI, but when she was still had the lab, I was a lab member. And so we were doing science at that time. And eventually she became the deputy director who would do all the office work, administrative work. And he joined as a co-worker. So we shared the same boss. So when he was traveling into China, Deborah said, you know, if you're going through Tokyo, why don't you meet Takeshi? And I know Takeshi very well. So we, one day, before, it was 2013, I think, so we met in Tokyo, and I brought him, and his daughter was also there, and we went to a underground chicken barbecue, a little place, and it's a smoky place, and then we talked about the future plans, and we just chatting, and then I think we should collaborate. And then we decided to have this agreement between the National Eye Institute and our Tokyo Medical Center, and this is the director of the National Eye Institute. But at the signing of this, we were not still in details what we're going to collaborate about. So after this signing, we went to my lab, and they decided, we talked a lot, and then we decided that we should, I thought we should work on Asian genetics. And he thought it was the same. He had the same thought, and that's how this Asian Eye Genetic Consortium sort of started. So this is the Asian Eye Genetic Consortium. I didn't know that the Asia was so big. I thought it was just around this area, but actually it goes all the way to Israel, and many countries are involved. And this is the goals. The first president of the AAGC was Dr. Prakash, and he set up some goals here. One is to share genetic information. Some of the countries cannot send out their samples out of the countries. So the sequence have to be done in each of those countries, but the data can be shared. So we want to actively promote this sharing of the data. Also, we need a sort of system to accurately find out what kind of mutation is going on. And this is basically exome sequencing, whole genome sequence, but still very expensive, but you can do this. And so the system to find out mutations. And also, another one is also cost-effective genetic analysis. And we also need to develop a database, which is becoming a most important project for the AEGC, to create a genotype-phenotype database. So number 4 is becoming sort of a center purpose of the AEGC. Also, we want to support collaboration between the Asian countries, and also want to organize meetings and seminars, just like we are doing the past 2 days, to promote this idea of collaboration together and share. So each country can then write a paper or do their own work, but after the submission, the data should be shared so that we can pool this data. And this way, if the drug company comes to us and say, usually the drug companies work on one particular gene, so if the drug company comes to us and said, how many patients do we have for this gene A? And we can quickly say, oh, in Mumbai there's like 3 families there, and in China there's like 5 families here, in Japan there's 1 family there. And then we can pinpoint who is the doctor who is responsible for each patient, and this drug company can directly go to the patient, access the patients, and start the clinical trial. So gathering all this information is extremely important. It's time-saving for the drug companies, so it's a very, very win-win situation, I think. So we started the Asian Eye Genetic Consortium 2014 during our meeting in Orlando, Florida, and the number is very small, and Professor Natarajan is standing right here, and I'm here. But next year, the Korean members have come in, and some more Japanese members has come in, China people has come in, and this year, a lot of people from Turkey, UAE, Singapore, and many other regions has gathered. And then next year, we're also going to have another annual meetings, and I'm sure it's going to be over 70 people of all the countries. And also, Dr. Prakash and I have been talking about what we should be doing, and we thought about publishing a book of AEGC. So this is a Springer publication, Advances in Vision Research, Volume 1. The title is Genetic Eye Research in Asia and the Pacific. This is the Volume 1 with 38 chapters. This book has just completed, and it will be coming out publication in March next year. And we're going for Volume 2. So if you're interested, if you want to write something, please challenge for the Volume 2. So this will continue as a Springer series of Asian Eye Genetic Consortium. So currently we have about 18 countries, and recently Iran has also involved. And this is the website, asianeyegenetics.org. So if you access this, you can get all the information. Also, this meeting with Dr. Natarajan will also be on the top page next week, I think. Yes. So with the collaboration between all the countries, what we're currently doing is to develop a genotype-phenotype, as I explained in a couple of slides before. And in order to do that, we need to have a common format. So currently in England, there is a institute called the Moorfield Eye Hospital, one of the biggest eye hospitals in Europe. And they have created a database 10 years ago, and they want to a little bit modify it now. Also, there is a database at the National Eye Institute called the iGene, and also they have started 10 years ago, but they are sort of closing down right now. There are, database also in China, and we also started making database 10 years ago. And so all these little countries have database, but it's not running as a common format. So it's a good time right now as Asian Eye Genetic Consortium to be involved heavily in creating database so that all the major facilities around the world can have a common format and share the data. And this will extremely accelerate the research. Now, once you have the genetic data, what does this really help the patients? So this is an actor, Angelina Jolie. She removed her breast because a mutation was found in a gene called BRAC genes. It's a very disease, a very severe mutation that is with a high penetration that she will have a breast cancer in the future. So she removed. And this kind of things is what we want to do. We want to detect the mutation before the disease will actually happen. And then we want to go beyond this, or to prepare, sorry, the English, prevent of not removing the breast. So right now we want to at least detect the mutation before the disease onset, and then go beyond that and to try to prevent disease onset. So for my thinking is the audacious goal for iGenetic is four, as listed here. First, we will share the genetic information, as I've been saying, and we will detect the mutation before the disease onset. In order to do this, and once this disease's onset has been, the mutation has been detected, we will apply the therapy, like gene therapy, cell therapy, artificial eye, that is emerging right now, and then probably it will become, one of them will become a major therapeutics of the future. And also, there are a lot of induced potent stem cells research going on, that the patient's cells can be reset to a stem cells, and then from there you can develop a retinal cells or even a eye cup from this. And this is becoming a major sort of a direction of the therapeutics. So I will just briefly introduce to you some of the therapeutics that is going on. This is the gene therapy vector. Usually, if you use a adeno-associated virus vector and you inject to the eye, this vector cannot penetrate the entire retina. So if you inject from here, it will come from here and it will try to pass through the retina, but usually it's stuck around this area. But this vector, called the 7M8 vector, has a mutated coat around the vector, so that when you inject from the vitreous side, it can go all the way down to the photoreceptor. This is a vector that is designed for the mice, and we don't have for the primates, but eventually we will have this specific vector that can penetrate the entire retina. In this way, we will have a very good therapy for all the diseases, retinal disease, most of the diseases, And many of the gene therapy trials are currently going on. I'm sorry for the little letters, small letters, but many trials are currently going on around the world. And one of the techniques is replacement of the genome. It's called the genome editing. So if you have a mutation, you want to replace it with the correct mutation sequence. So this is a technology called the CRISPR-Cas9 systems, that this CRISPR will detect a specific sequence in your genome, and it will cut the genomic DNA. Once it's cut, you will apply a normal sequence so you can replace with the mutations. And this is still the percentage of this replacement, we call it the recombination in the scientific words, is a percentage 0.8 to 1.8% successful rate. So it's a very low at this moment. However, in the future, I'm sure this will go up, and so you can target a photoreceptor, and you can replace with a normal sequence in these patients. And I'm sure the time will come for this technologies. Also, there is a significant progress in the stem cell research. This is the Masayo Takahashi from the RIKEN, who has developed retinal pigment epithelial cells from the iPS cells. And she has grown this into confluencies, and she will make into a sheet like this and transplant into the AMD patients. And she has already tried in 2014 September, and one patient, and she has completely stopped progression of the AMD. So these kind of studies is going on. And there's another study by Yoshiki Sasai. He has passed away 2 years ago, but he were able to make ES and iPS cells from an eye cup. So he's making a complete eye cup from his cells. And what he's aiming was, or he was aiming, is to replace the whole retina with cultured retina. Still a lot of challenge is facing, but these kind of amazing technology emerging, so combination of all these therapies, gene therapies, and artificial eyes, I think we will be replacing the current technologies. So in the future, I think this is the last slide. In the future, you will go to the drugstore, and then you will say, this is my sequence. This is my whole genome sequence. And then the drugstore, okay, you sequence this, and then try this therapy. So this happens. So this is a sort of a personal medicine that you have a sequence, and then your therapy comes out. So we're heading to this way. There are a lot of things to be done. But first, we need to find the disease-causing mutations. And that's what Professor Natarajan and I am trying to do for the aging. To find out the disease-causing mutations, and then we'll think about the therapeutics. So I'd like to thank all the people who have come today, and thank you for your attention.
Thank you, Takeshi. I request to give him a standing ovation for this first Dr. APJ Abdul Kalam Aditya Oration. Thank you very much.
Thank you.
Arigatou gozaimasu.